welcome, Miss Trishna Parasheri in our show called Rendezvous with Scientific Minds. Today, we will be talking about topics related to science, which causes interest and critical thinking in our viewers. That is what we have the idea of imbibing into the current generation. When the conversation is between two sides, we basically tend to decode the stuff which we are talking. Yes or no?
Yes.
So, before starting our topic, I would like to introduce our guest, Miss Trishna. She has completed her bachelor's in biotechnology from Guru Nanak Khalsa College and the Mumbai University. She has done a course from TC Science in communication skill course, done a course on cosmetology, and done an online workshop on chromatography. Interesting. Chromatography. Her work experience comprises internship program in Srijan Welfare Society for Mass Communication. And she has even taught in several classes as such as a teaching experience, as a goal of teaching experience. So over to you, Ms. Trishna. What we will be talking about, you can start with.
Okay. So first of all, thank you, Mahalakshmi, for inviting and as well as giving this opportunity. Like this is my first time having such a podcast. So today, like we would be only talking about related to my field, which is biotechnology. Like what is it and what it all takes to a biotechnology, and being a listener, like whoever will listen this, at least they would get certain idea like what exactly this field works like. Because like when I joined, I just joined for the namesake, like because I had to do IT, but I didn't got that. So I was like, घुस जाते हैं biotechnology में. But once I entered, I was like, there were many things which were very much new and very much interesting. So
Even when we face a situation like pandemic, it was biotechnology which came into use.
yeah,
Does
actually,
it sound interesting?
yeah, and at that moment, I was like, "Ki, I have to raise my collar. Like, I'm also a biotechnology student,
Yes.
but because of the vision, like my vision is towards B.Ed. So I was like, yeah, I can't do that much of in biotechnology. So I would try to inculcate my knowledge of biotechnology towards my studies or like wherever I will share knowledge.
That is an awesome ideology which you have, Miss Krishna. So, before diving into the topic, I would like to ask a simple question. What draws you towards science?
For me, it's like the realistic and a check which it gives, like like the proof material or something like that, and we can be like clear, crystal clear hota hai. If it's done, it's done. If it's not, it's not. So I always like in personal life also I deal things with like that only, like hai ki nahi hai. So science just approaches me and it just gives you sometimes, not every time, but sometimes it gives you a cooling, pleasant, like everything can be done under science
Yeah.
towards observation or towards inference. So
So we can collate it as a statement like it gives a logical reasoning behind every
yeah,
action.
yeah, exactly,
Yes.
exactly. Which everyone needs to get into it. Like everyone needs to get act, take that logical reasoning of science into their own life also. Ki logically, you have to take every decision.
Well, when we talk about the term biotechnology, it is like it sounds like a very bhari bharkam word when people we
Same,
see
same, exact
students.
situation. Like actually exact situation I got, like when I was standing in the queue in admission. I was like, my father was like, take in something, at least get admission. And I was like, that one uncle told me biotechnology. I was like, what is biotechnology? At least I should have certain kind of knowledge. So when I searched and when I got there, so now I know a virtual, like a whole deep meaning of biotechnology. But earlier for me, I was just decoding the word like biotechnology. Oh, something, the addition of
Something that
biology.
integrates life with technology, somewhat like
Yeah,
that.
so even I dealt with it. But when you go into biotechnology, it just shows you something like it deals with techniques. Like we biotechnology basically na, it deals with the live organisms. So that's why biology. So it uses the live organisms or enzymes of organisms to produce a new product using a technology, a kind of machine like PCR or what you can say like
RFLC, so
incubator.
many things are
Yeah,
there.
so many things. So to produce and process which would be useful for human life for us. So that's why it's biotechnology. And
Current,
talking
so
about
let us
like,
make things very interesting for current generations as we are propelling forward in our session. So
yeah.
let me start with a rapid fire round, Miss Krishna. I hope you are ready.
Okay.
Hmm. So let us understand the terminologies first. So whenever we talk about carbohydrates or whenever we talk about proteins, when we teach in our classes also, like and
Yeah,
for
yeah.
example, if we are teaching some students, teachers tend to directly jump into the terminologies. Basically they play with terminologies like proteins are there, this protein codes for this gene codes for this protein, but they understanding, baseline understanding is not there. So what are your viewpoints on that?
Exactly. We were, I actually had a very good set of science teachers. So, like, when we studied carbohydrates, proteins, fats, vitamins, and minerals, we were firstly told that there are certain things in our body which is essential. So first we were taught what is the meaning of essential, then essential nutrients, and then essential things. And for the body, we need some kind of essential things which would named as carbohydrate, proteins, vitamins, fats, and minerals. So after that, it decoded as ki ha, this function carbohydrate functions for this particular thing, proteins functions for this particular thing. So we were taught like this. And yes, basically, when in science, usually students does not have a keen interest. So teachers tend to directly go into the term because carbohydrates they link it to potatoes. So when children learn potatoes, they are like, oh, potato ke bare mein bol rahe. So it goes into a easy way of teaching.
You
So I
mean
think
to
that because
say
of
that
that,
we have to inculcate real life examples with the things which we are studying? That
yeah,
will
yeah.
induce more interest in current generation.
Exactly. And we in our school, like it was taught like that. Ki whenever you are doing science or any kind of like history, geography, anything you are doing, compare it with the real life one, and then you will better understand.
Next set of questions. Over to you, Miss Trishna.
Yeah, yeah.
Origin story of this branch.
So basically, it's like biotechnology was there before. For example, like I would give you like a certain example of the turning of curd from milk to curd, or from the rotten grapes to the wine. So this were all there. All process was going on, but people were not known. So I think in 1919 there was a sir named Caroli Eriki.
Correct.
He introduced a term which is biotechnology. Was like he biotechnology was present anciently with us, but we didn't came. So he gave a light to, and he did in agricultural department. I'm not much into it, but he did in agricultural department. So he's also known as the father of biotechnology. And later on, there was a scientist also. Herbert Bower, which like in our like first day of our biotechnology, we were asked like why you choose and all. And I think at that time when I was googling, so I also got like Herbert Bower was the person who gave a thing like for biotechnology. So what he did was everyone knew biotechnology आप use कर सकते हो in a normal way, but Herbert Bower give it a standard material like how you have to do it in a scientific proper manner by recombinating DNA or by gene splicing or by cloning. So that
Next
is the
question
origin
on to
story.
you now on genetic engineering.
Yeah. Genetic engineering basically, I would say, Malakshmi, like whenever I think genetic engineering will go as a process which you can use it in a laboratory based. Obviously, you can't use it openly, but you can use it in a laboratory based where your particular DNA, where a particular genetic materials will be changed or will be modified to make up a new organism or to make up a new set of DNA to fulfill demand or for the development. So
Oh.
it is basically a one type of principle. So there are two principles in biotechnology. Like one is genetic and one is bioprocess. So genetic deal with the genetic material like DNA, RNA and all, and bioprocess deal with like the chemical change for the growth of organisms like vaccines, antibiotics, enzymes, where you externally give it. So genetic is like which deal with internal side. So it is just host to say we are incubated inside our body.
Okay. Next question to you is, what is cloning? We have heard so many things regarding cloning and stuff, like in movies and in, TV series. People use this clone thing and make
Yeah,
a different idea. They put different ideologies in people's mind. That is, cloning is very easy and it can be done efficiently if you are powered or something like that. Can you clear that conception regarding cloning?
exactly, the basic meaning of cloning can be like a copy,
Hmm.
like a perfect copy, like how you take a photocopy or something like that.
Hmm.
So, but in biotechnology, it is like a specific organism or something must have that much kind of ability to multiply or to make a copies of antibiotic resistance gene. So it is definitely not an easy process. For example, if you take cloning for a recombinant DNA, you have to go through certain stages. Like first, you have to transform, like whatever um, organisms you have to work on. You have to transform the particular thing. Then you have to induce certain things which you have to make changes. Then you have to again transform it. Then you have to wait for that thing ki whether it is available to clone or not. Then you can, by using molecular cloning or by using PCR, then you can make a copies. So it is not a easy process. It is a very lengthy process. So
Even when I went through the process of cloning, when like certain examples I went through, I would like to share that with you. That is of Dolly sheep. It took so much of effort, so much of time behind that, but still only one copy was able to clone, and that was the exact copy of the mother, I think.
exactly. And what
Yeah.
happens in cloning is like you don't know the result would come out good or bad. So
Yeah.
it's like we are just dealing or we are just creating a scene like this. If we follow this method, there would be certain outcomes would be coming. But we can't rely ki that outcomes could be more one, two, three, or billion or trillion.
Yeah.
But in cloning, the matters is we are dealing with genetic material, and when you are dealing with genetic material, you have to be that much sensitive. So because of this, I think the Dolly Shipka cloning was done only once, and she created a history.
Yeah.
So
And I have even heard about this, that the cloned animals are not very healthy. Your viewpoint on it?
yeah, exactly. Because see, everyone has a inborn genetic material. What we are doing is we are recreating, we are recombining, like we are literally combining various whatever we need characteristics, and we are recombining it, and then we are hosting it. So obviously everything works like environment also, immune system also. So body takes it, body is ready, but to make or to go with a span, the particular organisms are not ready. So because of that, I think cloning has been very low. As you said, in many pictures, they show cloning as a very easy step, like one ka two, two ka three organisms.
Yeah.
Like I recently saw the Avatar movie, and in that they were showing like there will be many copies, and it's not an easy process. It's a whole lot through process which goes.
There is
So
still more way to go for us in
yeah,
genetic
yeah,
engineering.
that
That
is
we
definitely
can agree with, I guess.
definitely
Next question to you is,
yeah.
what are plasmids and vectors?
Oh, very nice question actually. Like vector basically in physics it's mean to show a direction to something, but in biotechnology it is a kind of a linker, like it links your organism towards the host. Like it is a kind of vehicle to carry your particular DNA segment to a host cell. So it just act as a linker. For example, I would like to I would like to give a common example where we can understand much better. Is that yeah, we can give an example of the Anopheles mosquito, as everyone might know that Anopheles mosquito is responsible for malaria. So here, Anopheles mosquito is a vector. So what Anopheles mosquito is doing is Anopheles mosquito has a parasite inside it, inside the body, which is known as malarial parasite. So the malarial parasite is transferred through Anopheles mosquito into the body of human cell. So it is a vehicle kind of or a linker for the parasite between a parasite and a human body. So that is what vector do. It just connect. So plasmid is a example of a vector. So like plasmid is basically nothing. It is just a structure. It is just a circular structure, a DNA structure. So what it does is it carries your particular DNA, like whatever modifications or whatever recombination you have to do, it carries it with the plasmid attack, like the plasmid attach, and then it is made. The plasmid is made to um, react or to get host with the particular cell. So
So
that is the
basically, what you
process which
mean
goes.
is whatever changes we need in a particular gene, that we recombine it, right? Whatever gene
Yeah.
which we
So basically,
require, and that
yeah.
gene we inculcate in plasmids. That are basically
Yes.
extra genetic material which is present in them in plant
Exactly.
cycle,
Where
and
it
then
is needed to manipulate your genes.
yeah, and then that plasmid we are taking and we are inserting into the organism of interest in which we need our
Exactly.
gene of interest, correct?
Exactly. Yeah, yeah.
Yeah. Next question to you is, what are restriction enzymes?
Oh, as the term only explains half of the whole thing. Restriction, it's like to stop. So what restriction enzyme do is, it is a kind of protein, like a kind of protein which is isolated from particular bacteria which you need. So what it does is it just cut or it just cleaves your DNA sequences at your specified site. Okay, and it produce a new set of DNA fragments with a known sequence at each end. So like for example, if I have to give you like if you have to cut a DNA ka double helicase structure, so you will need a restriction enzyme which is ECORI or E. coli ka restriction enzyme. It is
Eco
a
R1.
enzyme, yeah, yeah, equivalent. So you will need that to cut the DNA double helicase site. So restriction is it does nothing. It just cut your DNA fragments to like it just cuts and give you a certain amount of DNA fragments so that you can modify, you can
The
make the
gene of
changes in
interest,
particular
if
part.
we want, we will cut that and we will
Yeah,
inculcate into the vector. That
vector.
is the
Yes,
requirement for restriction endonuclease, correct?
yes, yes.
Okay. Now, since we are discussing so much related to DNA, RNA, okay, so let
Yeah.
us talk what is DNA, RNA.
See, DNA and RNA are basically for me is a protein. Okay. So
They code for
what
protein.
happens? Yeah.
They code for protein. The subtitles are going haywire actually. Oh boy.
I literally cannot hear your voice.
Are
Yeah.
you able to hear me?
What? Yeah, yeah, yeah. Now I can
Yeah.
hear you.
The
Yes.
subtitles are going haywire, I'm telling.
Oh, okay.
Just a
So,
joke. As I was telling,
yeah, yeah.
yeah, please continue, dear. Yeah,
Okay.
please.
So basically, for me, protein is like which is very much essential for your human body. Like very much essential. It just does a function like most of your function, like your hair,
Hmm.
your nails growing, everything is done by the protein. Um, but through RNA and DNA. DNA is the genetic material, like which I don't know. I thinking I'm getting a loss of knowledge right here. I have things clear in my
The
mind. I
thing is,
can't
it's
put
like
it
central
much.
dogma, we can say.
Yeah, exactly, exactly. Central
Whatever
dogma, and
protein which we are getting, that is from DNA, RNA, and then protein. It is transcribing into protein, right?
exactly. And DNA and RNA are responsible for the uh, from the start to the end, like even to make the chromosomes or to make an whole full organisms, you need DNA and RNA. Most probably, DNA is recombinant because like it does the most of the work. So I think,
Permutation and combination of
yeah,
these
topic
two
might
things.
not be clear, but
I mean, Arif, we talked so many, we covered so many things in such short span of time. You think it is really easy? No, I
no,
don't
no, no.
think so.
Like for me, protein, like protein would be a molecule which is made up of amino acids, like basically a nitrogen groups. So they are very much needed for the body to function properly. But in biotechnology, na, it is for me, I think, according to my knowledge in biotechnology, it is used for the production of gene expression.
Hmm.
Because what happens is from the protein you get the DNA and RNA, and you express or you modify the DNA and RNA, and then you make the changes. So protein is very much essential in biotechnology because if not protein, then nothing.
Nothing,
So
nothing.
yeah,
So there would be certain methods to, duplicate this gene and all that stuff. So can you show some
exactly.
light regarding the concept of polymerase chain reaction?
Yeah, for basically whenever you have to clone or whenever you have to recombinant, you have to literally go to certain kind of an process. Like first, like I said earlier, I might have said like a kind of transformation, transfection, and then non-bacterial transformation. You have to go like a three-step. First, you have to see which gene or which particular site you have to transform. Then what gene you have to induce, and then you have to let it make grow. But for a particular thing, like if you need a particular DNA or a particular specified DNA or a recombinant DNA, then you can use a method which is known as age, like the agarose gel electrophoresis. So the easy step would be you have to isolate some organisms, then you have to make it cut by using some restriction enzymes,
Enzymes.
and then you have to amplify it, which is here comes the cloning method. You have to make a copies, and then you have to insert like whatever modifications you need to have, you have to insert, and then whatever thing you need is obtained, then you have to just downstream the process, like you have to just make a copy or just make a thing like induce it into something host. For example, most of the time if you have seen in movies and all, we induce it in rat to see whether it's working or not. And towards if you are talking about towards very much related to the scientific term, then first certain material of DNA is added into a test tube, and in the test tube certain enzymes are added, restriction enzymes. For example, if I want to get a DNA from a plant, then I will add cellulose or lysosomes or I think a fungus ka bhi, there is something which is known as I think chitinase. Yeah, you can add that. And when you add the enzymes, what happens to uh, the DNA ka solution is that RNA is removed or any other materials is removed. So ribonuclease or everything is added, and after that your DNA is purified. Okay, so when, and I think the last process is you need to add chilled ethanol so that you will get a supernatant and
Strands
a
of
debris.
DNA
And after you
separately.
can get, yeah, and then you will get your required DNA as a thread. So this is how
That
you can actually
that was
isolate
an
a DNA.
extensive knowledge which we got. I hope all the questions were answered so diligently, and I'm happy with it. I hope you also enjoyed this session, Miss Trishna.
Yeah, yeah, yeah. I literally enjoyed. And with you know, like I don't know anyone who knows Mahalakshmi. With Mahalakshmi, you can go on and on. Like she fits the vibe. So
Oh, that's
I
so
have
sweet
to.
of you. Okay, Miss
Yeah.
Krishna, it was lovely talking to you, and thank you so much for giving us so much, giving us your time.
I also literally want to thank you.
Uh,
Yeah, and I'm literally apologizing if I missed something or
no,
I
dear.
may not be able to share that much of knowledge or like a proper way. So I'm just apologizing if
This
we
is
handle.
just an informal way of communicating, because when in recent times when
Yeah.
students interact with the teachers or any, they are not much very open, and they are not much very vocal about. I'm talking in general, actually. Not every
Yes.
student is vocal. So
Yes.
such conversations between subjectual topics help them to understand the concept in a very lighter manner, that will penetrate into their
Actually.
minds. So that is the main aim
Exactly.
behind this, which we planned out. So Rendezvous with Scientific
Yeah.
Minds might help thousands of children. I hope so in future. And
Yeah.
thank you so much, dear. This is Miss Pillai signing off. Take care.